All right, ladies and gentlemen, so today we're going to pick up with chapter 23, and we're going to really just look at what's happening to evolution at a population scale. So keep in mind that when we look at evolution in terms of how it works, one of the biggest misconceptions is at what level evolution is actually occurring at. And although individuals are playing a role in evolution in terms of what traits they're passing on from one generation to the next, overall, the smallest level that we can talk about actual biological evolution occurring is at the population level. So individuals themselves are not evolving because they can't control their genetic makeup. They can't change it. But population levels, those genetic markers, those genetic traits, can fluctuate from one generation to the next or over multitudes of generations because those individuals either die and don't pass on their genetic information by not reproducing, or they do reproduce, and so that they then contribute to the gene pool of the next generation. So what we're really talking about here is the combination of evolution and population genetics. We're studying evolution at a micro scale. Microevolution is defined as the change in the genetic makeup of a population from generation to generation. So we're studying genetic change in this population genetics over time. And really, this level of evolution is an interesting one to study because it's going to take the ideas of Darwin and descent with modification and natural selection and start layering on top of that Mendel's ideas of genetics, heredity. Both of these gentlemen worked on their theories, on their ideas, roughly at the same point in history, and yet they never talked to each other. They never communicated with each other. And so it took many, 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 many years before scientists started realizing that both the genetics and the evolution really were tightly bound to each other. And so that's what we're going to really look at, that connection here. So we're talking about this modern synthesis, right? This comprehensive theory of taking Mendelian genetics and Darwin's theory of evolution by natural selection and really giving us a way to start focusing on how populations change via evolution over time. So how do we define what a population is? Well, from an ecological standpoint, it's just a group of individuals in the same space at the same time that are of the same species. So we're looking at these localized groups. They're capable of interbreeding, which is often one of the definitions of a species itself, as well as not only being able to interbreed, but also producing fertile offspring. So that's a common definition for a species. But not only that, they are localized groups, and those members of that population tend to be slightly more genetically related to each other than they do of members of other populations of the same species. So when we look at something like the image here, all right, I've got two herds of these caribou, but they are distinct populations because of their genetic structure as well. So although they tend to be living in the same space, their genetic structure is just unique enough that even though they overlap, they are considered individual, distinct populations. So we're talking about their gene pool, right? From a genetic standpoint, from an evolutionary standpoint, we're really looking at this total collection of genes in a population at any given time. So we can consider all of their gene loci in that population, every trait that they are inheriting, but most of the time we'll kind of narrow down to a couple of those gene loci, or maybe even just study one at a time. But if we look at the alleles that are at that location, go back to remembering how we distinguish or how we identify genetic traits from Mendel's side of things, right? We use those capital and lowercase letters. So those are your different alleles, and those different alleles may contribute to being, say, brown fur or white fur. So if we have a situation where only a single allele, meaning only a single characteristic exists in the population, and there's no way to get heterozygotes and mix it all up, so they would either be all homozygous dominant, right, so 2 capital letters, or all homozygous recessive, 2 lowercase letters, then we talk about that dominant letter or that lowercase letter, the dominant allele or recessive allele, as being fixed. So that means that everybody in that population, that's the only trait that exists. Otherwise, we get these mixtures of alleles, and so you can have homozygous dominant, you can have heterozygous, you can have homozygous recessive, and they will continue to fluctuate from one generation to the next, but usually in a predictable pattern. So when we start understanding how genes get inherited and how nature then influences the inheritance of those genes, we can start talking about mechanisms that change allele frequencies from one generation to the next. Natural selection is going to be the only cause of that change that truly is about adaptive evolution, meaning that a certain trait, let's just say the brown fur, for example, is slightly more beneficial in that environment because of those environmental conditions than having the white fur is, then assuming this is a good, healthy population and natural selection is doing its job, then we should slowly see an increase in that brown allele with that white allele being reduced. That's what we're talking about with adaptive evolution. There are 2 other mechanisms by which we can change allele frequencies in a population that aren't necessarily evolution, and that's genetic drift and gene flow, and we'll talk through more about what those 2 look like over the course of this chapter as well.